Hello everyone, today in this video I am going to talk about the anatomy of dicot root and monocot root. So there is some difference between the dicot root and monocot root, and before starting the video I want to tell about the Histology theory of the dicot root and monocot root. So according to Histology theory, there is a four part of related to monocot root, that is calyptrogen, which is helping the formation of a root cap. Second one is dermatogen, which is helping the formation of epidermal tissue system, and third one is peribleum, which is responsible for the formation of ground meristem, and the last one is pleroma, which is responsible for the formation of vascular tissue system. But in the dicot root, according to Histology theory, there is only three regions. There is no calyptrogen, and dermatogen, peribleum, and pleroma. These three systems are well present in the dicot stem, and these are also responsible for the same thing as in monocot root system. So Histology theory is a well accepted theory for the organization of the root, and in this diagram you can see the major difference between the dicot root and monocot root. So starting with the diagram, you can see that in the dicot root and monocot root, in both type of root, in both type of stem, root hair is present on the above layer. So on the outer side, root hairs are present, which are unicellular in nature, and these root hair are uninucleated, ephemeral, and exogenous in origin. That is, these are not a particular cell; these are extensions of the outer layer, and these root hairs are unicellular, uninucleated, and and have very short lifetime. So below the root hair there is epiblema. You can see this is epiblema, and which is also known as piliferous and rhizodermis, and this is not covered by cuticle. But in monocot stem and dicot stem, this epiblema, or we can say epidermis, is covered by cuticle layer. But in dicot root and monocot root, epiblema is not covered by any cuticle. And below the epiblema there is a cortex. Cortex is parenchymatous in nature and well arranged in both dicot stem, both dicot root and monocot root. So till here there is no as much difference between the dicot root and monocot root. Now in the dicot root you can see there is a layer of endodermis, and this layer is made up of two type of cell. One is of passage cell, and the second one is of Casparian strip cells. That is, these in the Casparian strip, the cells have a suberin on it, and due to the presence of suberin, these are impermeable for water. But in the passage cell, these are thin layered cell, and there is no suberin in the passage cell. That's why these are responsible for the conduction of water, and these passage cells are opposite to the place of protoxylem. That is where protoxylem is present. At above that protoxylem there is a passage cell. Now you can see in the dicot root, below the endodermis, which is made up of two type of cell, below the endodermis there is a layer of pericycle, and in the dicot root, xylem and phloem are present radially. Means both xylem and phloem are present on different radius. These are not alternate with each other. These are present, these are not on the same radii. They are present on the different radius, and that's why there is a radial type of vascular bundles are present in the dicot root. And you can see that in both dicot root and monocot root there is a special type of vascular bundle are present. Means these protoxylem and metaxylem are of exarch type. Exarch type means these are centripetal in nature. Means protoxylem is lie toward the periphery and metaxylem present toward the center. So in the dicot root and monocot root, exarch vascular bundles are present, but in dicot stem and monocot stem, and endarch type of vascular bundles are present. This is main difference between root and shoot. Now in the dicot root and monocot root, in both xylem are of exarch type, and radial vascular bundles are present in dicot root and monocot root. You can see The number of xylem is diarch to hexarch in dicot root, but in monocot root you can see the number of xylem is very large in number. It can reach up to it can start from the exarch to polyarch. So in the dicot root only diarch to hexarch xylem are present, means their number is very less in in dicot root, but in monocot root the number of xylem are very large. It can reach up to polyarch. Now you can see in both cortex, in both dicot root and monocot root, cortex is parenchymatous in nature, but in the dicot root the the endodermis is of two type. The endodermis cell is of two type: Casparian strip, which is impermeable for water, and Passes cell, which is opposite to protoxylem and permeable for water. And in between the xylem and phloem there is a connective tissue, which is also known as conjunctive tissue. So in between the xylem and phloem there is a conjunctive tissue in both in both dicot root and monocot root. In the monocot root you can see, there is a number of there is large number of cells in the endodermis. And in the endodermis you can see there is a presence of suberin on the radial wall and tangential wall of the Casparian strip, and there is also passage cell opposite to protoxylem. In the monocot root there is a well defined pith present inside the vascular bundle. Pith, we can say pith or medulla is present in the center, in the central part of the monocot root, but in the dicot root there is no pith. There is no pith in the dicot root. So in the monocot root and dicot root there is a major difference in the pith. In monocot root pith is present, but in dicot root pith is not present. All other things are same as in the dicot root. Here is also Casparian strips and Passes cells are present, but the number of Passes cells in monocot root is large, because here the number of protoxylem is also very large. That's why in the monocot root number of protoxylem is that's why in the monocot root number of protoxylem is large. That's why Passes cells are also large, but in the dicot root number of Passes cell is very low. It forms. So this was all about the monocot root and dicot root. One thing is that endodermis endodermis have Passes cell, and these Passes cell help in the formation of lateral root. So in the monocot root number of lateral roots are high, but in the dicot root number of lateral roots are low, because here Passes cells are less, but here Passes cells number is very high. So this was a major difference between the root here, between the monocot root and dicot root. In the dicot root there is no pith in the center, but in the monocot root there is a large pith. Number of xylem is less in dicot root, but number of xylem is high in monocot root. Number of Passes cell is less in dicot root, number of Passes cell is high in monocot root, and these Passes cell help in the formation of lateral roots. Root here is unicellular, uninucleated, and ephemeral, and exogenous origin is in both, and epiplasma is not covered by any cuticle. Cortex is parenchymatous in nature, and Passes cell are opposite to protoxylem, and exarch type of protoxylem are present in both. Casparian strips are of suberin Casparian strips have suberin, that's why it is impermeable for water. So this was all about the dicot root and monocot root. I hope it will help you. Thank you, friends.